Can everybody hear me? Dr. Gupta, Dr. Amitabha Gupta, sorry, is a friend of my family, and so I'm very honored to introduce him to you all today. My first introduction to Guppy, which is how he's usually known, was during COVID. And I was invited to join a Zoom call that he was leading. And it was so great. I have a science background, and I was just hungry for the information. And he answered a lot of questions and gave out a lot of information. And I became in love with him. He's just great with science. So Dr. Gupta got his PhD at Columbia U in cellular, molecular, and biophysical sciences.
It's an easy pickup line.
That's a wow to me. And he went to Fred Hutchison's Cancer Research Center to do his postdoctoral work. And when he was there, he was looking to teach at a small liberal arts college. But Fred Hutch saw his teaching skills and kept him on. And so now he is let me get this right, the Associate Director of Scientific Content and Engagement at Fred Hutch. And he follows the ongoing research in 240 labs, so he can communicate to the communities and beyond and to us. So we are very lucky to have him. Dr. Gupta.
Thank you so much, Judy, for the kind introduction. And I'm really grateful to be here and talk with you all. I see more people coming in, so we'll give it another moment or so as they all come in. But what I'd like to ask for all of you is, I want this to be a conversation. If there's something you don't understand, if there's something that's weird, ask. If you have the question, somebody else does. So please don't hesitate to interrupt. If I don't make it to the end of my slides, I won't make it to the end of my slides. And that's perfectly fine. I'm just here to chat with you all about some of the really cool work happening at Fred Hutch and help you sort of get a little bit of insight into what we're doing and why it's so great to be at Fred Hutch. So, first question for everybody in the room. Who all has heard of Fred Hutch Cancer Center? Well, great. This is an easy audience. All right, I'm done. I'm going to leave. No. So, okay, I don't need to introduce Fred Hutch as much, but who am I, real quick? So I am, as Judy graciously mentioned, I came to the Hutch as a postdoctoral researcher to do some research, go on to be a teacher, and I ended up staying because the Pacific Northwest is gorgeous. How could I leave after coming here? Nobody wants to go to some quiet space somewhere, and it's where I've chosen to set down roots with my family, 2 dogs. That was summer 2018. This February, you might see the 2 of us biking around Green Lake as well, or taking walks, or getting into trouble, as we often do, the 2 of us. So I'm a scientist, a parent, a dog parent, a husband, and a community member of Seattle and the Pacific Northwest, just like all of you. And I'm really excited to chat with you all about what we're doing as Fred Hutch. So, very quickly, our talk outline for today, I'm going to give you a little bit of an introduction to Fred Hutch, because as much as you know Fred Hutch, it's possible you don't know everything about Fred Hutch. There's a lot going on there that is even a surprise to the people that work there, to be honest. I'll give you a bit of an overview of Alzheimer's disease, what we know, what we don't know. We'll talk about some of the Fred Hutch work in basic sciences on neurodegeneration with examples of research in worms and flies. I don't think we'll get to mice today. We'll save the mice for later. And a Q&A for anything that is unresolved, because, again, you will interrupt me frequently during my talk and ask questions, so we won't have any leftover questions by the end of it, right? Everybody in agreement? So if you see me, if you cannot hear me, just people at the back make the up symbol, and I'll keep an eye out for all of you. All the corners, specifically. Thank you. So, again, introduction, overview. So let's start with an introduction to Fred Hutch Cancer Center. So what many people don't know is that Fred Hutch was actually founded on a brother's love. So Fred Hutchinson was a great Seattle baseball player. So somebody's going to the Mariners tonight? Who's going Mariners? Yeah. He played for the Rainiers. He managed the Detroit Tigers, the Cincinnati Reds. But his brother, Dr. Bill Hutchinson, was an oncologist. And he had the misfortune of diagnosing his own brother with what at the time he thought was lung cancer. Turned out to be a different type of cancer later. And Fred passed away. But Bill was so committed to doing something in the realm of cancer because he was seeing so many people getting it that he established Fred Hutchinson Cancer Center. And there's been a lot that has happened at Fred Hutch over the last 50 years. So we opened our doors in 1975 and became Fred Hutch Cancer Center in 2022 when we merged with Seattle Cancer Care Alliance and became the adult oncology arm of University of Washington Medicine. And the thing that we're most known for, raise your hand if you've heard about it, the bone marrow transplantation. Who all have heard of bone marrow transplantation? Wow, great. Really easy. Who wants to explain it to me? No? Yeah, go ahead. That's exactly right. Well, I couldn't have said it better myself. As people with certain blood cancers, you get a treatment that wipes out the bone marrow cells that are making the cancer cells eventually, those stem cells, and replace it. And that was basically Dr. E. Donald Thomas perfected that treatment at Fred Hutch, for which he got the Nobel Prize in 1990. And one of the reasons why he was able to do it and other places weren't, because a lot of places were trying bone marrow transplantation way back in the 60s and were giving up on it. But he didn't, because he saw the curative potential of it. And he recognized the fact that he accepted he didn't know what he didn't know. So he brought together a team of experts, basic scientists, nutritionists, nurses, infection control experts, all of these multifaceted group of individuals who would approach the problem from their own perspective. And so this is his giant team at the time. BG's lunchbox down there. He brought together all these people, all these experts that were doing different things. And that's sort of been in the DNA of Fred Hutch ever since it was founded. This multidisciplinary approach, that humility to accept we don't know what we don't know. And we have to take on all problems and ask the most fundamental questions because maybe nobody's asked them yet. And you'll see, there's a whole spectrum of discoveries on this chart, including the work that we did to discover the cause of cervical cancer, leading to the HPV vaccine, the work we did with the Women's Health Initiative to build prevention work for postmenopausal women, creating immunotherapies that is reprogramming your own body's immune system to treat cancer. But what you'll notice is we also have 2 Nobel Prizes in fundamental science and basic research. So in addition to Don Thomas's work, our other 2 Nobel laureates are basic researchers. Dr. Lee Hartwell won the Nobel Prize to figuring out how cells monitor that they should be growing, whether they should be dividing, when they should be dividing, and those are mechanisms that are messed up in many cancers. So now those things that he discovered in the 70s are turning into targets for precision treatments for breast cancer. And Dr. Linda Buck was awarded the Nobel Prize because she discovered the science behind smell and how the neurons in the brain connect to one another to enable that sense of smell. We won't touch on her work, but there's a rooting in the work that we've done in neuroscience and the work around neurodegeneration. So I'll pause there before I move to my next section. Anybody have any other questions about Fred Hutch before I move forward? Oh, I'll take a sip of water while people are figuring this out. Great. So next, I want to move into an overview of Alzheimer's disease, what we know and what we don't. So a lot of people mistakenly believe that Alzheimer's and dementia are interchangeable. That's not quite accurate. Alzheimer's is a subset of dementia, and it's the most common cause of dementia symptoms, which is 2/3 of all cases. And dementia is the overall term for a group of symptoms that includes memory troubles, difficulty concentrating, struggling to understand, express thoughts, poor judgment, and acting impulsively, which, that's really hard because I do all of those things even now, not in my 40s, let alone, as we age. So it's a really hard thing to diagnose. But if we go further down, we think about it, there's about 7 million Americans who are 65 or older who are living with Alzheimer's in 2025. 74% of those folks are 75 or older. What we do know is that women are at an increased risk for developing Alzheimer's disease, but we don't know why. And Alzheimer's is a progressive neurodegenerative disease where you see a loss. So if this is what a healthy neuron looks like, you start seeing the loss of those healthy neurons and an increase in disease neurons. So a neuron, as I will use interchangeably, is one type of brain cell. So if I say neuron, I'm talking about one type of brain cell. We'll talk about the other type of brain cell in a moment as well. Everybody okay with this so far? Neuron equals brain cell, but it's also the nerve cells that go out all to our extremities and everything else. So hallmarks and what ends up being used to diagnose Alzheimer's is clumps in the brain, known as amyloid beta plaques, and alterations in a protein called tau. And there's just so many different factors that go into it that it's been really complex to untangle the effect of each one of them. And so you need these really large studies to figure out what's going on. But the work I'm going to tell you about today is taking it from a different angle. So you can do the large studies of collecting giant populations, figure out who has Alzheimer's, who doesn't, figure out genetic alterations, diet alterations, and try and make those maps. But our fundamental scientists are saying, actually, let's back this up. What does the end state look like? So we know that the brain shrinks. We know there's increased inflammation. We know there's blood vessel damage. We know there's decreased energy production, and there's creation of harmful molecules. So let's look at the fundamental biology of these brain cells, figure out what goes wrong to result in any of those other outcomes, and then maybe we'll get a guidance as to what is the driving factor behind it. So there's two approaches. So one is you look at giant populations and infer back. What our basic scientists do is they understand, how do things work? Now I poke it with a stick, what is that endpoint, and which of those looks more like Alzheimer's? Is everybody okay with that sort of setup? Yes, question. So which part? Yes. So these are the known age-related changes in the brain for people, yes. Other questions? Thank you for being yes, go ahead. That's an excellent question. Right now, the question was, can blood tests figure out disease neurons? Not yet. There are people that state that they may be able to, and maybe it's not blood tests so much as looking in your cerebrospinal fluid that's down your spinal column. It's not fully accurate quite yet, and part of it is to figure out what those molecules are, you have to know what those specific molecules are in Alzheimer's patients, but not in healthy patients, do the test in a general population, figure out if it's always predictive, but it's not there yet. But if you do this fundamental understanding, maybe you'll find those new markers that can lead to better and newer blood tests. Excellent question, thank you. So I'll pause there. Other questions about this state, because we'll start getting into a little bit more science-y stuff from the next slide on. Again, hydration break as always. So what is the work that Fred Hutch is doing on neurodegeneration, right? So going back to the fundamental processes. So the work I'm going to tell you today involves worms and flies. And why worms and flies? So the worm, which is called C. elegans, it's a type of worm, and the fly, which we use, is called Drosophila. The worms, the big plus is they have very easily manipulatable brain cells. We know which cells become the worm's brains, and we can just track them, and making a change in one, we know exactly what the endpoint is. So if we make those changes, we can map it back. With flies, we go one step more for other levels of understanding, like the connection between our gut and our brain, and the conversation between those two. Because one of the things that we've talked about leading to Alzheimer's is inflammation and obesity. That's why the gut-brain connection is really important for that sort of study. Again, we won't touch on the mouse today, but the mouse system is closer to humans. We know a little bit more, but we need to actually go back completely fundamentally. So let's start at the very beginning. We have a healthy neuron. Our researchers are asking, why is it dying? Very simple question. Well, we've talked a lot about neurons, but actually, neurons are not the only cells in our brains. Neurons only make up half our brain cells. The other half are cells called glial cells. How many people had heard of glial cells before? Yeah? How did you hear about glial cells? Yeah. Biology major, great. So what about you? Glial, G-L-I-A, glial cells. Yes, for glioblastoma, right. So if you've heard of glioblastoma, the origin cells are the glial cells, which is why glioblastoma, right? Yeah, question. Yeah, so the question is how much of our work is Alzheimer's and how much of it is... I think our fundamental researchers are just trying to understand the biology of the brain, and wherever that leads us, it could lead to discoveries for brain cancer and glioblastoma, astrocytoma, or it could lead to more understanding of Alzheimer's. They're taking an unbiased approach, so looking at the general biology, not restricting, to then figure out where it connects back. Because you don't want to restrict yourself and say, I'm only going to look down this one path, but ignore other discoveries that could benefit other pieces as well. But great question. Other questions? No? Okay, so glial cells, people weren't actually sure what they did for the longest time. Most people are like, they're part of the brain. They do something. When a neuron dies, we know there's a glial cell nearby that helps clean it up and sort of make sure that that space is opened up for a new neuron to show up. But that was all that was known about glial cells back in the day. So we had a new faculty join who said, I want to figure out whether the glial cells are just passengers or whether they have a more active form. And I'm going to use the worm because the human brain has, I don't even know what number that is for number of neurons, and an equivalent number of glia, and 1,000 trillion connections between each of them, which is not insignificant. There's a few to parse through. But thinking about those connections, they're all at various levels. So if you have a sensory neuron, so at the end of our fingertips, we have a nerve cell, a neuron, that touches the stove and figures out it's hot. So we have a sensing neuron at the end. The glia are around it to help stabilize it at the end. They also serve as the protector between the junction between a sensory neuron and an interneuron, which basically connects back to a motor neuron, which is the act of pulling your finger back, something that tells your body, move the muscle, get it back. So glia are involved in all of those processes in between, which is why it's so critical to understand what they do in this space. And again, they work similarly in C. elegans with a much smaller number. There's only 302 neurons and 56 glia in worms. It's a little bit more manageable than that. And let's see if this plays. Okay, over there, what you can see is a developing worm embryo. What's really great about that is right from the very beginning in a worm, we know from that first division which cell will become which neuronal cell. So we can make the change very early on and know that it will be replicated elsewhere. So it's almost like we have a pre-existing roadmap with the worms that then we can start playing around with. Because we know that there are different glial shapes, just like in humans. We know that they regulate neuron shape, we know that they prune them, and we know that they behave similarly in aging and disease in worms as in humans. So now, how do we make it, what are the types of tests that we do in worms? So there was a really interesting part of this work so the work I'm showing is from one of our faculty members, Akanksha Singhvi. She works in our basic sciences division on worms. And she took a really interesting approach. She found that there was this one molecule called KCC3, which, when it was present in the glia, it interacted with the neuron that sensed temperature. And when it was actively blocked in another part of the glia, it interacted with a neuron that helped the animal smell. So the same glia has the molecule sequestered in one part of the glia to help it support the temperature neuron, and it is an active antagonist in another part of the glia that helps the neuron smell. So what would you do with this information? What do you feel you would do with this knowledge and understanding? Changing a temperature, great idea. And if you're changing the temperature, you're hoping the worm can sense the temperature change. So what change would you make in these glia or neurons to see if the worms can still sense the change in temperature? Block? Kristen, I heard you say block something. I've got very good hearing. Yes, you would try and block KCC3 and see whether because it's only in the glia, it's not in the neurons. So you take that away, and lo and behold, the animal can no longer sense temperature, but it can no longer smell either. So it's actually involved actively to promote something, but that presence and that antagonism is also required to protect the neuron that allows it to smell. So it's a very subtle, it's playing two different functions within the same thing. And so when you go ahead and force it to move away from the temperature sensing neuron right around the smell neuron and have more of it there, you shut down the sense of smell. So with this system, basically, she can go to a single neuron glia connection and understand the biology between the two of those and really figure out what is happening at that one interface. That's cool, right? Questions? Yes? Mm-hmm. Yeah, that's a great question. The question is, how do we detect this? So basically, you take the worm egg, and for this experiment, say, the middle one, you find a way to remove this KCC3 molecule from one batch of worms. And so you have worms that have the KCC3 and the worms that don't, and you put them on a surface together where one side is heated and one side is not. And with a barrier between the two sets. And you see that the worms that have KCC3 will make it to the heat, turn around, and come back. And the worms that don't will just keep going because they can no longer sense the temperature shift. Cool, right? Great question. Thank you. I love talking about how to design experiments. Other questions? Yeah, please. A stock egg, yes. Yes, you want, so the question is, scientifically they have to be the same. Yes, you want everything the same between the two sets of worms, with the one exception that half of them don't have the KCC3 and the other half do. That's the only way you can tell this is uniquely involved in this one process. So they're called C. elegans, basically. It's a model organism that was, there was this one guy who was a genius in like the 1950s, 60s. His name was Sidney Brenner. He woke up one day and said, I need to find a new system to study X. I'm just going to use these worms. And there we have it. Yeah. Go ahead. Yes. There's modern methods and there's older methods. But one is you can find a way to, at the single cell stage, before the worm has started to develop, you can inject it with something that will actively block or change the DNA, so the information that makes that thing, or it will find a way that whenever the protein is made, so the DNA makes the code which is converted to RNA, which makes the protein, which is the functional unit. This, people can put in something that will start destroying the RNA, so no protein will ever get made in those cells. So, yeah, there's many different ways to skin that worm, I guess, if you will. Cool. So, what this showed that, yes, there are glial cells, but they're not just random passengers in a neuron's journey. They are talking to the neurons, and that conversation between the two of them is essential for the neuronal health. And so how active a participant are they, right? So we know that they can regulate and support neurons, but can they actively be the ones that instigate eating the neuron without the neuron telling them to eat? And so, okay, this video is going to be really hard to see and very quick, but what you're going to follow, so in purple is the glia, in green is the neuron. What you're looking for is bits of green in the body of purple, okay? So if a bit of green goes into the body of the purple, it means the glia are actively chomping on the ends of the neuron and digesting it in one way, shape, or form. So I'm going to play the video. Let's see. Tell me when you spot it. Those arrows, you can see right here and right here, there are bits of green being eaten away by the glial cells. So this was like brand new discoveries that the glial cells actively they don't have to eat away the whole neuron. They can just chomp at some ends and prune like a garden rather than take a full bulldozer to the neuron, just like trim the edges that aren't working quite as well. Do you want me to play again? Yeah. So you can see the green a little easier on the left side in just this space without the purple. So I'm going to play it one more time, just because it's so fun. But we needed some really high-powered microscopes to be able to see that. And those, they were donated by a very wealthy donor who was really helpful to this project. And without that donation of the microscope, we wouldn't have been able to get this resolution of data as well. Right, okay. So basically, what this data is showing is that glia aren't just wiping neurons, they can be pruning little bits and ends. And maybe as we age, it's incorrectly pruning bits and ends, reducing the function of the neurons as we age. And so what you can see, and so what Akanksha and her team did was they went into the glia, figured out the junctions at which the neurons were being pruned, and figured out which molecules were present in a high level in that space. And what's really interesting is I'll point to, anybody recognize the word Parkin? Yeah, it's named because it's been associated with Parkinson's disease. So there are factors that are known to be associated with an outcome, but nobody knew what they were doing. And she has found through her examination that Parkin in glia, not neurons, which is where everybody was looking for them, play a role in breaking down those neurons. And so the origin of Parkinson's disease as well may be on the glial side, not on the neuronal side. So people may have been looking in the wrong place. And without this fundamental understanding, we wouldn't actually get to that point. So her big takeaways were that glia pruned dynamically, it alters the animal's behavior, that it can't sense the temperature, it can't sense the smell. And so there's more to be uncovered from there. I'll pause if anybody has questions. Yes? Yeah. So, yeah, excellent question. So is Michael J. Fox and Parkinson's working with us? So I know that Akanksha and her lab members have submitted grants to that foundation. So what we also, a lot of our faculty, because again, funding constraints are really high right now, they're forced to apply to multiple foundations beyond the federal government to get research funding, and that would be one of the places they apply. But there's a lot of competition for grants right now as well. So it's not certain that they will get it. Other questions? Nope? All right. So far I've shown you the idea that glia can regulate and support neurons, and they help degrade dying neurons, trigger regrowth, prune them. So let's come up with two models of how glia and neurons, what sort of interaction can lead to neurodegeneration. So there's two possible models. Can somebody come up with one? Ideas of what could be off between the glia and the neuron interaction that could lead to neurodegeneration. Didn't think there would be a quiz component to this, did you? Well, I'll give you one, let's say. So one hypothesis is the glia breakdown. And so there's nothing else left to support the neurons, and over time, the neurons will die away. What's another possibility? Based on everything we've talked about with the glia pruning the neurons dynamically. Yeah. They're pruning, they're overactive, they're pruning too much, exactly. So the idea is that it's either not there and the neurons are dying, or it's overactive and it's pruning all over the place. That's now leading to the death of the neurons. So it's mispruning. So how do we figure out what the difference is between those two? Which of those is the right method? And it's possible, and spoiler alert, I'll tell you at the end of this, both can be right depending on the circumstance, but these are two possible models by which neurodegeneration occurs. Right, pause for other questions, because I also need to hydrate. Yeah. So, sorry, can you repeat your question? So all our researchers are actively going to scientific seminars, symposia, conferences, and talking with others about their work and sharing their work. So a lot of people do end up collaborating around the country, or people will find the published materials and want to work with our faculty. So we have a lot of collaborations around the world, around the country, around the world, but also within the campus, which is actually really cool. Like it's very rare, and one of the things that was so unique about Fred Hutch, which made me want to stay, was our faculty have a real open door policy, and they like to work with one another and share information with each other. We have a lot of publications that have many of our faculty together, versus often on the other coast, it will be just one lab's work, and people just sort of hoard the data in the hope of a higher impact publication. Yeah? No, nothing's off topic. Yeah, excellent question. So does the overforming of glia have anything to do with the formation of a glioblastoma? It is fundamentally possible. I was reading some material so again, glioblastoma is glial cells that are growing out of control. So yes, there's a lot of glial cells which should normally be regulated in a 1-to-1 ratio with neurons, that you start getting more and you start forming this mass. And there's something about glioblastoma where they form the glial cells actually form their own network independent of the neurons that is keeping them alive, which is a very weird part of the biology. But so the next phase of that will be thinking about how do the glial cells talk to one another and exchange information to start getting at that question. But great question. Other questions? So to try and suss out which of these models is right, I'm going to switch model organisms and move to the fly, the Drosophila. And so one of our faculty members, her name was Akhila Rajan, she came to Fred Hutch with the question of how do the fat cells in our body communicate with our brain cells to let us know that we're full? That's it. Because at the end of the day, we all need a certain amount of energy, and our body knows what we need. How do we know when we are full, when we have enough energy? And so she said, I need an organism that regulates how much it eats very well, and that makes it really hard for Guppy to say fat fruit fly many times. So she started working with fruit flies, because you never see a fat fruit fly. And so using that as an organism to think about how do fruit flies know that they're full enough? What is the signal that goes from the gut to the brain? And can we exploit that? And that was her entire question. But in uncovering that, she has made some discoveries that help us parse this model of whether one leads to the other. And this is what's so beautiful about basic research at Fred Hutch. Two different people are exploring two completely different mechanisms for different reasons. But as you explore the fullness of that work, you start seeing where things connect and how one person's work can help another, and how that science builds on science, and the importance of getting people who ask the questions regardless of an endpoint, because you don't know where we'll end up. So that's what's so cool about being at the Hutch. Okay, so what did she find out beforehand? So her previous work found that there's one molecule that's released from the fat gut of flies that goes up to the brain that signals fullness. The fly is full, it is done. And so altering that one molecule allowed her to say, actually, I can trick the fly into doing what Guppy does and eat like 17 donuts for breakfast in the morning and just keep going down that route, right? Or go to a bar and watch a game before he has to give a talk, one of those things. You know, how do we make that? So now that she knew the molecules, she's like, I'm going to make a change in that, see what happens. So what was that impact of that overfeeding on the neurons? So to do this, she created a system where she could measure insulin in the brain. So insulin is something that goes in and moves sugar in and out of the cells that need it. And so if the insulin receptor is too full, it won't allow more sugar to come in. So what does that do for the brain cells? So what she found was that this was to figure out that so HSD means high sugar diet. The flies that had a high sugar diet or got rid of the molecule, so they ended up overfeeding, did show that the insulin receptor was tapped out and the fly was not taking any more glucose in. So this was her marker to say, right, I found the right cells that are not working as normal or they've eaten too much. Now let's figure out the downstream consequences of that. You with me? So green means that the glucose transporter is working fine. The flies have a normal diet. They're able to take in the food. They're able to function normally. So what happens when the function is compromised? So this is the color means something different. The green here means the glia cell are ready and functioning normally. So she found a marker that states that the glia cell are ready to eat and do things. And when you move the flies into the high sugar diet or create insulin resistance, the glial cells are no longer functioning normally. Their state of readiness is gone. They're not available to start knocking down unhealthy neurons. So what does that mean for function? So this was a really interesting and cool experiment. So basically the idea was that they took flies with either the normal diet or the high sugar diet, so in purple, and they injured a neuron, and then they watched how the fly recovered, whether the glia were present to function normally and keep the ends pruned and keep it normally set up, because you don't want a broken neuron to sort of keep persisting, because if it's broken and it's taking up space, you want it to get it out of there and cleaned away so the new neuron can grow. So here is a healthy cell. So there's the same neuron on two sides. She injured one side. Nothing grows. It's been cleared away. Whatever was injured is completely gone. Versus in the flies that were fed with the high sugar diet, the glia are no longer functioning to prune away the broken pieces that you can see down here, because the bits of the neuron still exist long after the cut has been made. So something about a high-fat diet has compromised the glial cell ability to function normally, leading to an increase in unhealthy neurons. Yes? So extrapolate to your brain. I don't want to, because I don't think we've gotten that human data yet to confirm. But what this suggests is that in inflammation or obesity or insulin resistance, the ability of the glia to function normally and prune correctly as needed or to clear away debris is reduced. And so in obesity and high inflammation, there's just a lack of proper glial function, which could lead to more neurodegeneration. So basically, again, what her data is suggesting is that as we go in and have unhealthy diets or lead to high sugar diets, we are compromising our glial function. Data I will not show you, which is still too early to be published, is some work with the mice, which is exploring whether sort of infections can lead to increased glial function, which has the opposite degradation. So two parts. One is the stymieing of glial function through diet, or the overactivation of glial function through infection. But that is not ready to print yet. That information is not fit to print yet. So with that, I will pause and open up for Q&A for anything unresolved that people may have, and it can be about this talk or anything else about Fred Hutch that I'm happy to answer. Yeah? Yeah, excellent question. So the question is, how long did it take to be discovered? So when Akanksha came to the Hutch, it was 2019. At that time, we didn't even know that the glia had an equal function as the neurons. But that was published about 4 or 5 years later after she showed up. Yeah. Question? Right. Yes. So that's the final end stage. The end stage of Alzheimer's is the idea that you have these plaques that are building up. But what leads to those plaques building up, what leads to the neurons stopping working, are there proteins that should be cleaned up that are no longer being cleaned up that stick around? That's sort of what we're getting. We're trying to figure out further back of what was the origin that led to the plaques eventually. So the plaque is proteins that should have been cleaned up? Correct. Can you say that again, please? So people can hear what you said. Yeah.
So the plaque just oh, so plaque that's associated with Alzheimer's are proteins that did not get cleaned up by glial?
We don't know. Officially, the question is, was it not cleaned up? But we just know that there's an accumulation of bad protein. But what leads to that, that's the question. And maybe this is one of the mechanisms. So, questions?
Does the lymph system have anything to do with this?
Does the lymph system?
The lymph, the other word, close to lymph.
Lymph? Lymph. So lymph is the system that moves blood around our body.
Yes, there's
Yeah.
the lymph system.
I actually don't know the answer to that question. But something I can look up and I can get back to you. Yeah, question. Yeah. Yes. So that is a terrible question for them to ask. But the fact of the matter remains that there have been really interesting studies out of other countries that show improvement in cognitive health with an increase in hobbies, connections, partnerships, friends. So the more you are actively using your brain, the healthier you are. And so comparing like brains of people in other countries where they have active social lives versus places in America where people end up being more lonely, there's a huge difference in brain function among those different groups. So what you all are doing, you're helping your brain function. Yes.
Molly, let me bring this to you. I have read, well, first of all, how does the United States compare to other countries in terms of the rate of dementia? And because I have read where Japan, for instance, has a very low rate of dementia. And do I correlate that with the fact that they don't have the high-fat diet and obesity? Is that what I'm getting here?
So correlation does not equal causation. Let's start there. So the fact that they don't have a high-fat diet and they don't have obesity and they have lower neurodegeneration, those are all like facts that could be correlated, but we need to figure out whether those are linked one to the other, and it is causative. So lots of things can move in the same direction, but whether one leads to the other, we have to actively prove that factor. Yeah.
Mary?
Yeah. Question, the microphone coming?
Can you say that the relationship between what you're describing and high sugar, high fat diet is pretty certain?
So the data shows that high sugar, high fat diet does lead to reduced glial function, reduced neuronal pruning in flies. That's about as far as I will go, because that's what the data says. Until we do the experiment in other organisms and other things that we can, really carefully make those changes, I don't want to say, but it gives a new opportunity for somebody else who's working in other systems that are closer to humans to make those exact adjustments and see what it does.
Thank you.
Yes. Sorry, science does move slow sometimes, but at the same time, we want to make sure that we are saying the right thing because as you know today, it's so easy to find whatever information you want out there. We want to be absolutely nailed on before we say anything definitive. Yeah.
What is a burning question you would like to know?
What is the burning question I would like to know?
Yeah.
Good question. I want to. Okay. Here's we got, and this is sort of an invite to have me back in a few months again. So the burning question that I have is that we humans are complex organisms. It's not just us. There's as many bacteria and microbes living on us and in us as we have our own cells. So our microbiome, which is the bacteria and viruses that live in our gut, on our skin, there's as many of those as there are cells that are human on us. So what is that interaction between the two? And specifically with the gut and the brain, because our gut has as many neurons as our brain does, and those are in close proximity to the microbes. So what is that crosstalk with our diet and our microbiome that could be leading to depression, anxiety, neurodegeneration in that space? So the microbes are my big, fascinating question. Next.
Are you working on those?
No,
I
am happily retired from the bench. I have not had an experiment fail in 8 years. I am 100% successful in everything I do, which is great, unlike most scientists. So before we go, I'll say I would love to stay connected with you all. So you can learn more about Fred Hutch at fredhutch.org/campaign. Talk to me afterwards if you want to join a Fred Hutch event. We have a lot of those. There's not many coming up in the summer, with the exception of Obliteride, which is our run, walk, bike ride, where we always need volunteers, lots of volunteers, because it's a massive event. If you want to donate, you can go to fredhutch.org/give, or talk to my friend Stephanie Henderson over there. She's a gift officer with Fred Hutch, and she knows way more about ways to support Fred Hutch than I do. I'm just the science, she's the giving. I will turn off the mic in case people have questions on the quiet side. So thank you so much, all of you.